it is quite different. Normally I come up with the hypothesis and I figure out a way how to test that hypothesis. You guys kind of came to me with your hypothesis, and together we devised a set of experiments that we thought could test your hypothesis, which was that twist inhibition would promote hair growth in androgenetic alopecia.
All right, guys, before we get into the interview with Dr. Claire Higgins, I just wanted to give you an overview of why we're running this Twist1 study for hair growth in the first place. Typically, we would ask the researchers to give us their hypothesis, but that doesn't make very much sense in this case because the HairDAO community fully designed and ideated this Twist1 study. So as just a brief overview for the uninformed, Twist1 is the second most significant hit in genome-wide association studies, GWAS studies for androgenetic alopecia. It also sits, it's kind of a hub gene, if you will, or it's a transcription factor. And so it touches a lot of the other genes which we've identified in terms of being really important for hair loss. So that includes our spondins, that includes FGF, that includes TGF-beta, ID proteins, and more, ultimately cascading down to when Twist1 is elevated, it suppresses the Wnt pathway, which is super important for hair growth. While we don't put a ton of stock into mouse studies, Twist1 inhibition has been found to essentially stop progression into the catagen phase on mouse hair follicles, which is also, super encouraging. Twist1's main functionality in the human body seems to be during embryogenesis for sort of bodily structure formation. So think the formation of, your sort of bone structures, particularly the skull. There's actually a condition called Shaitri-Chotson syndrome, whereby these people are missing one copy of the Twist1. And what you see in those patients is that they have misshapen or misformed skulls, but they actually have very full, thick heads of hair with very low hairlines, which is obviously interesting for our purposes. Later in life, in adulthood, Twist1 actually, elevated Twist1 seems to have very few positive effects and is actually associated with a bunch of nasty conditions like cancer, like metabolic issues, like fibrosis, none of which seem very good to live with. So our real thought process, our real goal here is that by suppressing Twist1 in the scalps of older people who are already fully formed, their skulls are already fully formed, their bone structures are already fully formed, not only could we regrow their hair, but we'd also decrease their chance of suffering from a bunch of the conditions I just mentioned. So, which obviously gives this the best chance of ultimately making it into humans outside of just regrowing hair, decreasing the risk of a lot of other conditions. With that said, I'll stop talking and we can get into the podcast with Claire. All right, we're here. This is Hairy Matters episode 9 after a long hiatus, back by popular request. And I'm here with Dr. Claire Higgins, a very highly anticipated guest by everybody in the Discord server. She's been running our Twist study for about the past year now. And so the purpose of this podcast was sort of to get to know Claire a little bit better, learn about sort of the work that they've been doing so far in the lab, and then also where we're going. Thanks so much for coming on, Claire. And I think just to start out, it might be kind of interesting for the community to kind of just hear sort of how you got into hair follicle research in the first place and sort of just how you got into this very niche but interesting field.
Well, first, thank you for having me. Thanks for inviting me on. I've always been interested in hair. So I obviously did my PhD on hair and have run through since then, which I presume your viewers know. But actually, what a lot of people don't know is my grandfather had alopecia universalis. So he lost all his hair when he returned from the war. And it was thought to be related to a stress event. And so in all of my life, when I knew him, he had no hair on any of his body. And my gran always used to say, that's why you got into hair, because you're... I don't feel that was the main reason, but maybe it was a contributing factor. Like, I've always found hair really fascinating in the complex relationship that we have with hair. So when I was an undergrad, so this was around 2002, I went to lectures by a professor called Colin Yehoda. I was doing natural sciences, and this was a developmental biology module. and he gave a lecture on how they had like relocated bits of hairy skin from one body site to another and induced new hair growth. And it was these experiments that had actually been done by his postdoctoral supervisor, Daniel Drelly, back in the 1970s. And I just found that style of experimentation really, really cool. I was interested in science, but I didn't really know what to do. And I always was telling everyone about this amazing lecturer. And then he offered a PhD position in his lab. And one of my friends was like, you know, that guy that you're always talking about, well, he's got a PhD position, you should apply for that. So I applied for it, and then I did a PhD on hair development and also exogen, which is hair shedding. And then sort of the rest is history. I stayed in the field. I did my postdoc researching hair. I researched alopecia areata, androgenetic alopecia, also trichomegaly in my postdoc. And then I started my own lab in 2014, and it focuses on both skin and hair development, regeneration, disease.
Nice. No, yeah, we were actually doing a Twitter space the other day with Dr. Bloxham out of New York, and he was very excited to know that we were working with you. So your sort of reputation clearly precedes itself. Now to sort of go on to the topic that everybody's super interested in, this study we're running with you at the Imperial College London, focusing on sort of the top gene targets that the community has identified, TWIST1 and TWIST2. This was, I guess, probably a unique sort of project for you, because we sort of came to you and we said, like, we have this target, we're really excited about it. We sort of have this study design that we'd really like to try out. You have a lot of expertise. You have a lot of expertise working with both occipital and frontal follicles, which is also, really interesting from our point of view. A lot of people test on occipital follicles, which don't really miniaturize. So that's, probably a little bit lacking across the broader field of research. But yeah, I think just a starting point could be interesting for people to hear, sort of what we've done in the study so far, where we are, and then how you would interpret sort of the data or results so far, and where we're going next.
Well, I should just start by saying I'm a reader in the Department of Bioengineering, and so it's slightly different to the US system. My primary job is to teach, and I'm funded because I teach undergrads, and then on the side I run a lab. And as you say, it is quite different. Normally I come up with the hypothesis and I figure out a way how to test that hypothesis. You guys kind of came to me with your hypothesis, and together we devised a set of experiments that we thought could test your hypothesis, which was that twist inhibition would promote hair growth in androgenetic alopecia. So first we wanted to, I guess, you've come up with a list of compounds and silencing RNAs that you think will inhibit twist expression. And then we need to make the links to whether twist inhibition actually promotes hair growth. But our first question was, do the compounds even promote twist inhibition? And so we set up an experiment with dermal papilla cells in culture, and we put either your compounds or your silencing RNAs onto the dermal papilla cells, and then looked at how effective they were at reducing twist expression. So we did PCR to look at gene expression and immunofluorescence to look at protein expression for all of the different compounds. And we also checked if the compounds were toxic. Like, it's no good if they reduce twist expression, but they kill all the cells in the process. So we did a live dead analysis to check for toxicity, narrowing down. And we also looked at a variety of concentrations of each of the compounds and looked at the optimal concentration that could reduce twist expression without compromising efficacy for toxicity, right? And that's the first question. Can we even inhibit twist expression? And we can. The silencing RNAs, of course, work the best. They reduce expression of the gene and subsequently the protein. And they're not toxic at all, which is really nice. The compounds are quite variable. A lot of them show efficacy, but then they're also quite toxic. And so we have to select, for the studies going forward, slightly lower concentrations. So we're not killing off all the tissue, but we don't know if that's going to really impact their efficacy. And so the next question, which we're moving on to now, is does twist inhibition have an effect on hair growth? For that, we're using it's called the Philpott model because it was devised by Mike Philpott when he was at Unilever. But people also call it like a in vitro hair growth model. In most labs, they just go, oh, I'm using the Philpott model, and everyone knows what you're talking about. So we're using this in vitro hair growth model. For that, we take occipital follicles from patients who are undergoing hair transplantation. They give their consent for any follicles that are not used during the transplantation process to be used for research purposes. And that's all approved by ethics committees. And so if they give their consent and there's follicles left over at the end of a surgery, we get them shipped. And then we can culture them in this particular growth medium that sustains their growth for around 10 days. And the idea is after the first day of growth, we'll add in different compounds to try to reduce twist expression, or the siRNAs to reduce twist expression. And then we'll look at a couple of things. We'll look to see whether we actually did get twist inhibition in this ex vivo hair follicle, because it's quite different to cells. We don't know whether the compounds are actually going to get into the areas that we want it to get. And we'll also look at hair growth rate. So we'll see whether we can accelerate the daily rate of hair fiber production in this assay. The follicles can spontaneously go into a regression state at around day 6. So another thing we can look at is if we can extend the amount of time that they stay in the growth state before going into this regression state. And so they're the questions that we'll be asking in this second part of the study.
Got you. Nice. And remind me, so are in this next phase of the study, is it going to be primarily occipital, or will we be mixing between occipital and frontal hair follicles?
It's quite difficult to get the frontal follicles. We do have ethical approval to get 15 frontal follicles and 15 occipital follicles matched from patients, but that's a separate kind of route. For this first study, we're just going to be using occipital follicles because they're ones that are discarded and not used in transplantation. Whereas when you take frontal follicles, obviously that's we're taking samples specifically for a project. They're not being discarded, right? So we go via 2 different routes. So for this first study, where we test all 10 compounds and silencing RNAs, we will just use occipital follicles. We'll see if there is an effect. And if there is no effect, we might put DHT onto the follicles to induce regression, and then see if there's an effect that we can prevent that regression. So we kind of have to figure out what the baseline is first before we then try to augment it. And then, because it's harder to get the frontal and occipital follicles, we're going to use this process to kind of narrow down our selection and select 3 compounds going forward that we will then test in the frontal and occipital follicles matched from the patients. No one's really looked at frontal follicles. We don't even know if they go into catagen maybe at 4 days instead of 6 days, or whether there's differences in the way that they respond to DHT. Maybe they have different growth rate. So we have all those questions to ask first, like establishing the baseline. And then once we know the baseline, we can put the compounds on and see if they have an effect. So there's 2 different outputs though, right? One is, do they reduce twist? And the other is, does reduction of twist have a functional effect on hair growth?
Right. And so for sort of this first stage of the study, where we're really looking at sort of toxicity and then also like the ability to inhibit twist. And also, by the way, if I don't know, like you want to just name drop any of the molecules, then they're proprietary, whatever, we can bleep them out after the fact. But I'm curious, just based on what you saw, if so far, if you're seeing any sort of winners in terms of I guess finding the sweet spot, but behind sort of maximum twist inhibition and lack of toxicity.
I mean, for me, the winners are the silencing RNAs, which is maybe not what you want to hear because you want to identify a compound, but they're very effective. There are some of the compounds that do inhibit twist, not to the same degree as the silencing RNAs, and so that would they would probably be the winners. There's some compounds that are very toxic. So even if they inhibit twist, they're not ones really to take forward. But actually, I think at this point, it doesn't really matter so much. We need to test the original hypothesis, which is that twist inhibition actually has an effect. Because there's no point like sourcing other compounds until you know that, the inhibition is going to have an effect. And so really we can do that with the silencing RNAs, right? We can effectively knock down twist and we have a measurable output. And if we then see that inhibiting twist does have an effect, we can try the compounds that inhibit it to a lesser degree than the silencing RNAs, and they might still have a partial effect. Or we can try combinations of compounds that, might act in a synergistic manner to inhibit twist more if twist inhibition is really what we need to do. So we can answer it first, and then if we need to find other compounds, we need to find other compounds, or it might be that the inhibition that we're getting from the compounds is enough. We also don't know that.
Yeah, and I think that makes sense, right? It's sort of, now that we know in the cells that we can inhibit the twist 1, twist 2 with siRNAs, it's like, let's just see if that actually has the effects we want on the hair follicles before we go through the whole hassle. Were you at all surprised? I mean, I guess to the positive side, that even the siRNAs were as effective as they were at inhibiting twist. I know that a lot of the feedback we sort of got around this is because twist 1 in particular is a transcription factor, that it could be really hard to bind to at times. Or was this just sort of like, I guess siRNAs are pretty predictable and they're designed exactly for this case?
Yeah, I wasn't surprised with the siRNAs. I mean, we expected them to work, and they did work at even quite low concentrations. But I was surprised that we got inhibition with some of all the compounds did inhibit to a degree, some more than others. And that really surprised me. I don't know how you selected the compounds, but some of them actually do inhibit twist quite well. You expect it with the siRNA. It's designed specifically to target the gene, right? Your compounds are not. So that surprised me. That was interesting.
For the compounds, we were primarily drawing, honestly, around drawing from a lot of sort of experimentation that's been done with sort of just like people trying things on themselves online, reading through those servers. And then also we did sort of some computational biology work, sort of a broader screen to sort of predict a couple molecules that we thought had a good chance. So yeah, I think you probably have sort of an interesting case of how you typically source your hair follicles. So maybe if you just speak to sort of the process of sourcing the human tissue, and then also just sort of, what the ramifications you think might be if we could speed that feedback loop up a bit.
So we get our hair follicles from patients who are undergoing hair transplantation procedures already. And we have quite a lot of relationships with different hair transplant clinics around London and also actually in Europe. And the patients have to sign a consent form. They have to give written approved consent that they consent for their hair follicles to be used for research purposes. They also get a patient information leaflet which tells them what their follicles are going to be used for. We're not going to extract their DNA and find out things about them. We're going to use their follicles in the lab. And the consent forms and the patient information leaflets are all approved by a research ethics committee who oversees it. I'm quite lucky at Imperial. We have an Imperial College healthcare tissue bank, which you can use to store and use human tissue in different projects. We have 5 hospitals associated with the university, and so that's why they have this central tissue bank to kind of coordinate tissue that is used for research purposes. And so when the patients give their consent, the follicles are routed on paper through the tissue bank to my lab, and we have approved research projects that we're allowed to use the follicles in. And the tissue bank is regulated by the Human Tissue Authority. They have like a license to use human tissue there. So it's all very straightforward. The limiting factor is like we get our hair follicles from patients undergoing hair transplants. We work with several clinics, so sometimes we get two lots of follicles from two different clinics on a day, but there's like 10 people in my lab all wanting follicles. And even with the amount we're getting, they're still fighting over them because it's not enough. But like a surgeon can only do one transplant a day, right? So I don't know how we would speed up that process. We just have to be a bit strategic. And sometimes I have to step in and say, you use the follicles today, someone else will use them tomorrow. You need to share. Yeah. It's so valuable for our research. Like, we're so grateful to the patients who are consenting for their leftover tissue to be used. Otherwise, it would just go in the bin, right? And we use it for so many different things. If the tissue is if it wasn't good enough to be transplanted, sometimes it's a bit kind of chewed up, but we can still isolate cells, we can set up cultures, we can do digestions. Like, there's a lot of different things we can do with the tissue. So we also take a look at it and then decide what project it's going to go to.
That's a PSA to all the people getting hair transplants out there, is to pay it backwards and contribute to the research so future people don't have to go through the same fate. Yeah, so okay, so moving on, just I guess back to the next phase of testing, and I think we spoke about this sort of briefly earlier. But are you going to be using immunohistochemistry for the readouts when you suppress twist on the in vitro follicles?
We're looking at RNA to look at gene expression, and we'll also be using immunohistochemistry or immunofluorescence probably to look at protein expression of twist itself, but then also of other things. We'll look at things like caspase-3 to see if there's apoptosis in the follicle or cell death at all. We'll look at Ki-67, which is a marker of proliferation. Hopefully we get more proliferation, less caspase-3, a few different markers like that. We can also go back to the follicles, because once you've finished the experiment, we embed them in this compound called OCT, which kind of preserves them, and then they go in the -80 freezer. And later on in the study, we're going to do RNA sequencing of twist-treated follicles to try and identify the downstream targets that twist inhibition leads to. And so if we identify some interesting downstream targets and we start getting to the mechanism of how this is occurring, we can go back to those follicles in the freezer at a later date and then do immunofluorescence or PCR on those follicles. Probably immunofluorescence, because we'll have got the genetic data or the gene data from the RNA-seq.
Right. They will want to know where it's expressed.
It gives you a bit of spatial information.
And so just, I guess, in this very, like, this, whether it's immunofluorescence, IHC, you mentioned a couple of the biomarkers, but in terms of like what success looks like. So if we're probably going to start with sort of the siRNAs to make sure that I guess to give ourselves the best shot of inhibiting twist in the hair follicles. What are sort of the main biomarkers that you would look at and say, like, oh, we upregulated this one a lot or downregulated this one a lot. This is, very, very exciting for potential hair growth.
Most people look at you have your follicle, and the fiber comes out at the top. So when you start the assay, you chop it with a scalpel. So there's a clean edge against the top of the follicle and the fiber which is coming through the follicle. And then each day that fiber moves out of the follicle a millimeter or so. And you can measure, we'll take daily images of the follicle and the fiber that is being produced. And so we can measure the growth rate of that fiber. That, for me, will be a key output. When you put DHT on follicles, it's supposed to slow the growth rate and also make them move into this regression state. And so really I would be looking to see if twist inhibition prevents the slowing of growth rate induced by DHT. On a normal follicle, it might increase growth rate over normal, but we're not really then asking the question of what's the effect of DHT, right? So I think we want to see what DHT does and then see if we can prevent that effect.
Gotcha. And so this is maybe a bit of a technical question, but I'm interested that you sort of weight the actual sort of measuring and growth of the follicle in the well that highly, and it makes sense. One issue that we've had, I guess, at least when we first started doing the Philpott model here in New York, is that sometimes when we first started, the hair follicles were sort of like at an angle in the medium, if you will. And so the measurements would be inaccurate. So do you just sort of like make sure they're pressed up against the bottom of the well to ensure sort of consistent measuring? Or
No, on the first day when you set it up, I always make sure it's not floating. I would push it to the bottom, and once it's at the bottom and it's lying flat, if it was like at an angle like this, you can't take a photo. You need to push it flat, but you don't need to squish it. If you squish it, you'll damage it and it will go into the regression state. I would just get a needle or something and gently prod it so it would lie flat. And sometimes it rolls over, but you kind of want the orientation of the follicle to be the same every day. So I normally even manipulate it a bit to get a good angle for taking the image.
Yep, we learned that lesson the hard way, but it's so I think it's just important that we have you clarify that.
Well, it's important as well a lot of the follicles can get damaged in transit or get nicked a bit when they get taken out of the scalp, and they will quickly go into a regression state, but you don't, or they might not grow at all. And so actually it's best to set up the assay and then wait, go the next day and see what ones are growing and what ones are not, and then start your treatment on day one.
So for the next question we had, and maybe, you tell me this might fall a little bit out of your realm of expertise, but I'm curious, just, we love this target, and you're supportive of us loving this target. But I'm curious what you think the biggest sort of scientific or technical challenges might be in terms of translating sort of the findings from this study into an actual treatment that could go into people's scalps.
So I guess there's two different aspects to this question. I'll address more the technical side first. So I guess this is the issue you have with any drug discovery pipeline, right? A lot of drugs that you come up with computational models and you predict the drugs will work, and they work in cell culture models, and then they don't work in the clinic. And that's because the models are not that effective at predicting, or cell culture models in general are not very good at predicting efficacy in a clinic. And another one is toxicity or off-target effects, which we can't really assess right now. You can't really assess that until you get to the clinic. I'm hoping we've sort of mitigated the first issue with our experimental design. Maybe not with the cells and culture. They're so different to, tissue. But by using human follicles in a lab, we're actually testing on the same end organ that we want to test on when we move to the clinic. So I'm hoping that the efficacy issue will have been addressed. But then we're also putting Twist directly into the media and it's soaking into the follicle. I think the biggest challenge is actually going to be getting it to target the follicle if you're rubbing it on the scalp.
We have a topical dutasteride study going on exactly for that reason. Sometimes delivery, like also particularly to the bulb versus going like fully systemic or sort of just getting stuck on top of the epidermis can be.
Yeah, and ideally you want it to be topical because then you lower your off-target effects, right? And then you don't have to worry so much about the safety side of things, because that's another big issue. Drugs either fail because they're not effective or they're toxic. Normally because the models that are used to look at efficacy and toxicity are not that effective. But yeah, hopefully using human tissue actually has circumvented some of this.
I do actually have a quick follow-up on that, because we often think about the hair follicle being actually like a really great preclinical model, particularly when you compare it to sort of like single cell or mouse models. I'm curious, just like in your experience, have you seen sort of the Philpott or just like the, human hair follicle organ culture model? Have you seen it be more predictive of in vivo environments than single cells? Like, is that real or not so much in your experience?
That's quite hard to answer. I haven't seen much actually be translated to the clinic. So it's not really that comparable. I mean, I went to an amazing talk yesterday on glioblastoma, and they were using single cell technology to identify like therapeutic targets for that. And of course, it was a presentation, so they're very positive about what their output, and they've chosen what they're going to include on their slides. But actually, the symposium itself was more about spatial transcriptomics. The issue with single cell is that you don't have any spatial information. Current issue with spatial is that you don't get the good resolution. Like, I think it would be really, really fun to do spatial transcriptomics on follicles with twist inhibited. That would give you spatial information and help you identify the mechanism, see whether actually what's going on. Yeah, each one by itself, like just doing the Philpott model is great. We learn about hair growth. It probably is quite translatable. It's just things haven't really made it to the clinic.
We need more people trying to translate things. Yeah.
Single cell allows you to ask different questions. It's really good for identifying therapeutic targets. Like you can analyze cell ligand interactions and see how different, all the different cells are acting together. But you need to rebuild up that information to try to get a spatial view of the follicle. So maybe, moving forward, I mean, spatial transcriptomics is becoming a really hot area to work in. But the technologies, like a couple of years ago, they were terrible. The resolution that you would get, I mean, for one spot on your chip, you'd get 20 cells. It wasn't subcellular, but now it is. One
well was practically like an entire hair follicle.
Yeah, it was the entire DP. I looked at the beginning. I thought, we'll do spatial on dermal papilla. And one well was the entire follicle with the entire dermal papilla.
Yeah, it's like a retro video game or something like that.
Yeah, because it's only about 100 microns, right? And yeah, it was not very good. But now you can get subcellular resolution.
No, we're definitely exploring something in that realm in live time, and obviously think it would be creating a map, if you will, of the hair follicle for everybody to sort of reference.
I think it's really like these technologies are moving so fast, and I don't want to get left behind, right? But some are not appropriate for the question you asked me at the time. Like we looked into spatial a few years ago. As I said, it wasn't suitable, but that doesn't mean we can't use it again when it becomes suitable, right? So you just have to keep on moving with the technologies. Whenever I go to a presentation, I always think, how can I use that for my research? Like, how can it's not always about the results. They're interesting. It's always about, their techniques. And I think, how can I apply that to my question?
And then I guess before we go into community questions, I think just something that's fitting, because you are sort of one of the most respected people in hair research in general, and we have but traditionally trained, and we have a lot of untraditionally trained people in our Discord server who are actually obsessed. And so I just a quick couple words on sort of any advice and guidance you would give somebody looking to get into hair loss. I guess the best way to sort of start tackling the subject, because it is pretty broad.
Let the science guide you. I think that sounds quite cliche, but you're doing the research because you don't know the answer. And I remember when I was a postdoc, actually, I had a PhD student who had just joined the lab come, and they'd read this paper which they had taken as fact. And you have to take everything a little bit with a pinch of salt. If someone does one thing, it doesn't mean you shouldn't repeat it and check it. It doesn't mean that that's gospel and things don't work by a different mechanism. The hair follicle is so complicated. There's a multitude of different pathways all working together, lots of signaling interactions between the kind of different cell types that are present in the follicle. Hair growth is quite a complicated thing. It's not going to be dictated by one molecule, except maybe androgen receptor. If your target doesn't work, look somewhere else, or think that things could be appreciative that things can work together. Might be a synergistic combination of different things that are working together. But don't go into it thinking, I'm going to test this one thing and I'm going to prove that this is the answer. Like, start off with a hypothesis that you want to test, but take it step by step. And if you get a different answer, that's not the end of things. You just need to take a slightly different approach. So it takes quite a long time to be quite thorough. But yeah, be adaptable. Don't take what's written as proof. I mean, test it yourself, and things going to work through different mechanisms, right?
Absolutely. I mean, yeah, there's so many different kinds of hair loss as well, right? Who's to say, I don't know. Obviously, alopecia areata and androgenetic alopecia are different. Telogen or fluvium, totally different. I think our server is most focused on androgenetic alopecia, but I think generally the more you can learn about the follicle in general, maybe the more conclusions we can come to.
Yeah, and actually, like, a lot of pathways can work on different types. So JAK inhibitors have been developed for alopecia areata. They've been approved now by the FDA, the EMA, different drug enforcement agencies for treatment of alopecia areata. But there is a publication that shows that JAK inhibitors can kickstart the bulge when it's kind of in a dormant state, which would work for androgenetic alopecia, right? So the same pathways could also work for multiple different alopecias. And if twist works, that's brilliant, but that doesn't mean that you stop there, right? It means you find something else that also works, and then maybe that in combination with twist is even better.
So in your opinion, are there any sort of other genetic or epigenetic targets that you think could be worth looking into even more, potentially funding?
Well, for that, I think we need to think about the mechanism by which the follicle miniaturizes. So there's two aspects to miniaturization, right? One is a shortening of the anagen phase, so you get very short hair fibers. But the other is the dermal papilla becomes dimensionally smaller, and the dermal papilla width directly relates to the width of the hair fiber that is produced, and so consequently you get a very tiny hair fiber being produced that is also not growing for very long. So it's a combination of those two factors that actually leads to androgenetic alopecia. And pretty much everybody is focusing on the first one. So they're focusing on things that prolong the anagen phase. And there is reason for that. The follicle only gets dimensionally smaller when it cycles. So in anagen, you've got quite a big dermal papilla. It goes into catagen. You get this kind of movement out of the dermal papilla, so it becomes half the size that it was in catagen and telogen. And then at the start of telogen, you go back, or the end of telogen, you go back into anagen, and you get this movement of cells into the dermal papilla, enabling it to double in size again and go back to its original size. You never get a follicle in anagen that suddenly the dermal papilla shrinks and it starts producing a really thin fiber, right? So the shrinkage must be related to the cycling. So if you can keep a follicle in anagen for longer, don't let it cycle, you're going to prevent miniaturization. So that does work. If it is going to cycle, the other way to look at this would be to try and identify drugs that stop that shrinkage occurring during the cycling. And if you can find that, then it wouldn't matter if it went into catagen or it cycled, right? And no one's really looking at that side. No one's looking for drugs that like increase the size of the papilla or alter this kind of movement between the sheath and the papilla that occurs during this transition. And that's because we don't really know what we're looking at. And there's not really a good model for that either. If we're doing human research, we have the Philpott model that I told you about, but that enables you to look at this anagen to catagen transition. We don't have a human model that allows you, at the moment we don't anyway, we don't have a human model that allows you to look at the telogen to anagen transition. And that's kind of where the difficulty lies. So for testing, it's very hard to test. I mean, you can put a compound onto dermal papilla cells and see if it makes them proliferate. Lots of people do that. And they then say, this means that this will make the dermal papilla bigger. But as we discussed earlier, like these cell culture models are not representative of what's going on in vivo. So I think thinking about the target is a good way to go. Like we there's two targets, right? If we can identify drugs to target the other side of it, then we've got the kind of full package.
Right. And what kind of things in your opinion would be sort of, I guess this might be getting into trade secrets here, but what kind of like is it really just like upregulation of Wnt signaling like in the dermal papilla, or Do we just not know, or
No, I don't think we know. I don't think it's like Wnt regulates the cycling, but I don't think Wnt is controlling the size. There's some old studies that looked at, cell adhesion molecules. There's a large number of cell adhesion molecules in the anagen dermal papilla. You lose them during telogen and catagen. So maybe it's that, right? Is there a loss of kind of DP identity, so the cells just don't want to go back there because there's nothing telling them to go there? We know what can control the transition, like we know FGF7 promotes telogen to anagen, FGF5 promotes anagen to catagen. We don't know what controls the size.
I do think there's some hope that whether it's, second, a third, fourth order of magnitude effect, because something like twist one impacts so many.
Yeah, it might also control the size, but we don't have a way of testing it, really.
Yeah, 100%. Yeah, exactly. I know, just because I know some people will be shouting at the screen like it has the potential to sort of grow the dermal papilla as well. So I think I know that is a hope, but yeah, it is kind of funny, right? I guess the only way to know.
We can and we will look at that in the Philpott model. So you can also look at the size of the papilla, but it's within anagen. So we can see if it makes the anagen dermal papilla a little bit bigger. So it is a measurement we can take, but it doesn't mean that it's going to affect that, influx or efflux during the cycle.
Right. And impossible to make that assumption of what will happen in cycle.
When you test it clinically, you might prevent miniaturization of kind of existing hairs, which would be great. But even better, maybe you would start reversing the miniaturization of already miniaturized hairs.
What's the? I'm blanking right now. The period of like actual sort of telogen. How long is that period?
So in non-androgenic follicles, it's about 3 months, and in androgenetic alopecia, it can be years. They get stuck. So there's one aspect is they lose the volume, right? You have this shortened anagen, and you also get a prolonged telogen. So the cycle kind of shifts.
Yeah, so I'm almost wondering if even in a clinical setting where it's sort of like a 6-month period, I'm wondering how much of that cycling would actually get caught even in the.
Well, you might kickstart the cycle, right? Maybe all these follicles that are sitting in telogen, maybe you'll kickstart them back into anagen, and you'll see hair fibers coming out from sites where there was nothing growing before, and because they were in telogen, and maybe the telogen hair had fallen out. And then over time, maybe they'll get bigger. We can learn a bit. I mean, I know one of your community questions was on TRPS, and that does control hair size. So, we don't really know how. So it was difficult to answer other aspects of that question because I don't know how. But the literature does suggest that when TRPS is mutated, you get smaller hair follicles. When there's position effects on TRPS, you get larger hair follicles. There's sources of information, and I guess, can you package them together and figure out a way to test the hypothesis?
On to the community questions, and the very first question that I have here. We know the epithelial bulge hair follicle stem cell population remains intact in AGA. What about the mesenchymal component, especially hair follicle dermal stem cells? Could their depletion or a shift where they reside, in parentheses, proximal distal, be a critical factor?
We don't really know where they reside in humans, so that's a little bit difficult to answer. But yeah, I definitely think they're affected. The bulge cells do remain, but their ability to their ability to differentiate into progenitors is affected in AGA. So you have less progenitors, more bulge, and there's less of this kind of differentiation. Now that could be because there's an intrinsic difference in the bulge cells that is associated with AGA. It could also be that they're not receiving the right signal, and that signal could come from the mesenchyme. We know it comes from the mesenchyme during hair growth. Why doesn't it come from the mesenchyme during pattern hair loss, right? So I think the mesenchyme definitely has a role. If you are getting this increased cycling that we've discussed earlier, or like shorter anagen and the follicle is cycling more, every time that occurs, the dermal sheath stem cells need to repopulate the dermal papilla. Occipital follicle, they might need to do that 20 times in a lifetime. But in a miniaturizing follicle, maybe they keep on having to repopulate the dermal papilla, 60 times because it keeps on going through the cycle. And so eventually that might be beyond the lifespan of those sheath cells. They might just get exhausted. I have a hypothesis that AGA is a form of premature localized aging just on the frontal scalp, because the cells are having to do much more than they would have to do on the same hair follicles on the occipital scalp. So I think the mesenchyme definitely has a role. And it might be that this, increased cycling or the mesenchymal signaling to the bulge is altered. Maybe it's not to do with age at all, but it's just related to their ability to signal. If we can recreate the signal and get the bulge cells to start differentiating into their progenitors again, then maybe you could reverse the miniaturization. And maybe the bulge cells signal to the dermal papilla and get them to expand in size. We don't know where the signal for the dermal papilla comes from either. So I guess it's communication between the two, which is needed.
So I guess what I just heard that all the baldies will appreciate is that you believe that reversing miniaturization is a possibility.
Yeah, absolutely. It's not fibrosing alopecia, right? There's no fibrotic tissue which is impairing cell movement or impairing function. We just need to figure out what the signals are that get the dermal papilla to expand in size again. It knows what it should be, right? There's studies in mice that show when they ablate the cell number in the dermal papilla to reduce it, then the hair follicle cycles, then the papilla always goes back to the original size that it was before the ablation. It has some kind of size memory, and it's that size memory which has been lost here. It doesn't know what size it needs to get to, right? And because it is not getting to the correct size, there's impairments in its signaling, and there's also impairments in the bulge cells in that they're not receiving the signal to differentiate and form the hair fiber. Yeah, but there's no scar tissue or anything. Like we just need to recreate the right signaling network. I absolutely believe it is reversible.
You believe that even in the case of somebody who's been bald for years?
Yeah, this is different condition, but with alopecia areata, people with universalis who had it for 20 years, when they went on JAK inhibitors, they regrew all their hair. They'd been bald for 20 years, right? You never get past the point of no return. The stem cells don't disappear. So long as your stem cells are there, you can just need to reactivate them.
So then I think another community question, which I think is a great one because you've done more work on this area. But do you still see dysregulated dermal papilla cell influx or efflux as a key factor in AGA? Can we rule out dermal papilla cell apoptosis or identity loss as the primary cause of reduced dermal papilla size?
I think we can rule out apoptosis, but I don't think we can rule out identity loss. So apoptosis comes after efflux. The cells in the papilla, in the transition from anagen to catagen, they migrate out of the dermal papilla into the dermal sheath. So you reduce the size. So that's the efflux. And then once they're in the dermal sheath, they then apoptose and the dermal sheath is lost. But the dermal papilla cells themselves don't undergo apoptosis. So I think because the efflux precedes the apoptosis, I would say that the efflux is more important there. Whatever goes out is going to apoptose, and what you get left with is your dermal sheath stem cells, which are the only ones remaining at the end of this. No, I think identity is really important. As I said before, the papilla has this predetermined size that it gets to, and that's part of its identity, whether it's because there's a load of cell adhesion molecules in it, which are, causing cells to go in and stay there, or whether it's because there's a signal within the DP, again, recruiting, or whether the DS cells know that they need to migrate in at the start of anagen. That's all related to the identity of the DP. And it could be any one of those things. We don't know which one it is, but that for me is what is lost.
I do know that people are very interested in sort of Takashi Suji's work and hair cloning. So really just like, are you optimistic about this approach? Do you think that the sort of organoid hair models will get to a point where they can approximate like normal human hair growth?
I think with Suji's work, it's a great model. I don't think we'll get to the point when you can transplant those follicles. But I was saying earlier, we don't have a model to look at, something that increases papilla size. But maybe something like this could be really useful as a screen to identify therapeutics that can augment the size of the follicle. And then you don't need to transplant the follicle. Or maybe you transplant your tiny follicle, but then you put a therapeutic on to make it get bigger. But I don't see them making a large enough follicle to transplant to directly replace, lost follicles. I see it more as a screening tool or identifying therapeutics.
That's interesting feedback, that it could be really useful in terms of studying that anagen to catagen progression, and then the sort of life cycle of the follicle, how it sort of changes in the catagen telogen phases. Do you think are you less excited about it from an actual standpoint of implanting them back into like a human scalp, just because the hair follicle is so complex?
No, I just don't think you need to. I think it's a lot simpler. Why do we need to make this whole follicle outside of the scalp? The body knows how to make hair follicles. It did it originally during our development, right? The idea behind hair cloning was that you would take dermal papilla cells out the occipital scalp, transplant them into the frontal scalp, where they would induce an entirely new hair follicle. And we never saw that happening. But I think there's still a lot of merit in hair cloning. Now they propose that a transplantation of dermal papilla cells will augment existing follicles. And I think that's a lot easier to do than try to make an entirely new hair follicle. If the dermal papilla can restore its identity, they can migrate into the papilla and enable it to expand in size, right? So all these things actually need to come together. I think the cell injection stuff is really good. I don't think we need to transplant the whole follicle. I think we need to augment the ones that are there, or get the body to, make it. But the ones that are being made in the lab, they're useful for other things, right?
Our study with you is just one of many studies that you and your lab are running, and one of many hypotheses that you guys are analyzing. So more broadly, I think everybody would just love to hear what you guys are focusing on right now, what excites you most about hair research in general, or even outside of hair research.
So I've got 10 people in my lab right now, and I think 3 of them work on skin and different fibrosing skin diseases and wound healing projects, and then 7 work on hair related projects. So we obviously have the project with you on Twist One. Other people in the lab are looking at augmenting hair follicle cycling. Some people are looking at hair pigmentation. Others are looking at evolution of hair, why we lost body hair on our, why we became the naked ape, supposedly. I'm quite interested in patterning. So why, which is one of the reasons for the evolution project. Why do we lose hair only on our bodies? Why do we get long hair on our heads? And then why on our heads does hair miniaturize, but in a specific pattern? So we have another project on the genetics, or the genes that discriminate between frontal and occipital follicles that might contribute or explain why we have this pattern. So a variety of different things. But I really, that's all the projects I've just listed are very intrinsic to the hair. In recent years, I've become really interested in how the hair, I believe the hair controls everything. So like hairy skin heals faster than non-hairy skin. Hair does everything. So all of the projects I just described are intrinsic to the follicle, but actually in recent years we've become really interested in how the hair kind of augments the skin. So we've got some projects on hair follicle remodeling of scar tissue, hair interaction with the sensory nervous system, which obviously is in your skin, and how the hair follicle has a role in touch sensation, things like that, which really excite me. And I never know what the answers are going to be, right? These projects are just all quite exciting, and they reinforce to me every day that the hair follicle is really interesting and that it's a great model system to work on.
I guess one last thing, which could be kind of funny to talk about. So I'm assuming your lab receives a lot of sort of traditional funding and very like mainstream academic sources. I'm curious just to hear your experience in terms of how it's been different to work with HairDAO on a study versus sort of your more typical sources of fundings. Maybe the pros of doing a study with HairDAO, the cons as well. Like we can talk about that. We can talk about the cons also. But just the full spectrum, I think, would be great to hear.
So I do receive funding from charities, grant agencies, but actually they don't really fund a lot of hair research. It's always been kind of a bit of a joke for my colleagues that a lot of the hair research I get funded is through companies, and when I get funded for other projects, it's like the skin or the wound healing projects, I get it through the grant agencies. But either way, if the funding I've got through companies has still been set out by me, which is quite different to the HairDAO project, where you came to me with the hypothesis. That was a first. Normally I come up with the hypothesis. I think this would be a cool area to work in. What questions? And we sort of write a research proposal and say we're going to do these experiments to test this hypothesis. And then you need to argue that the hypothesis is worthy of testing. Whereas you came to me with the hypothesis and it was quite different. You said, how would you test it? So we'd remove the first step in a way. And it was quite specific. We've got a planned route and we're sort of doing those experiments, whereas sometimes companies come to me or I pose more open questions. Like, what is the role of X in skin biology? And then we'll do some experiments and it will lead to a completely different route, which we then can go off on a tangent, right? And that is what the benefit, I guess, of grant funding. It's much harder to get. You need to write a really long grant and it takes a year between writing the grant and potentially finding out you had it and recruiting someone. But it then is quite flexible. Like if the initial hypothesis doesn't prove correct, you move on to a different route. So I quite like that aspect of it. But then with you guys, it's quite set experiments. But it was interesting because it was a different way for me to approach something. I had to come up with the experiments to test your hypothesis, which was quite fun in a way.
I figured. And no, I mean, to be honest, it's like the way we work is that you could come to us with sort of a more open-ended study and it would of course go through that layer of that layer of internal debate. And this idea that we came to you is one where sort of had a couple champions in our Discord server who were fighting for it over the course of a year at least, and then also sort of designed it themselves and everything. And so we were like, okay, this is something that somebody had already fought for, and we needed sort of an expert to sort of execute on it.
That's kind of why my lab is sort of a bit of a niche for this in a way, because we have questions that we're interested in, and obviously we're able to do them, but not without the tissue. The clinicians, a lot of them are very engaged in research and have really interesting research questions they want to ask, but they don't have the facilities. And that's kind of your position as well at the beginning. And now you have this lab, right? But when we first started this relationship, you didn't. And so you had the interesting research question, but you didn't have the facilities. It's the same as the clinicians. They provide us with the tissue. We can answer their interesting research questions. You provide us with some funding and we source the tissue and can answer your interesting research questions. And it kind of is just this synergistic relationship in a way.
And now we're going to have you guys come to New York and teach us everything. All right. Well, thank you so much, Claire. I think this has been an awesome conversation. I think people will like it. And looking forward to sort of the next phases of the study. So thank you so much.